Species continuity is reliant on two factors: genetic stability and genetic variation. Mitosis and meiosis ensure the continuity of species by maintaining a balance between these factors. Mitosis is preceded by interphase. Interphase is broken up into G1 and growth phase, where the cell grows larger and duplicates organelles, S, where DNA is synthesized, and G2, where error correction occurs. In the first mitotic phase, prophase, the chromatin condenses into chromosomes, with each chromosome consisting of two identical sister chromatids joined by a centromere. Also, the nucleolus disappears and the nuclear membrane breaks down, enabling microtubules to form. Then metaphase occurs, where spindle fibers from the centrosomes fix to kinetochores on the centromere and align the chromosomes along the metaphase plate. Anaphase follows. Here, the sister chromatids are pulled to opposite poles of the cell by the contraction of spindle fibers and the cleaving of proteins in the centromere. During telophase, the daughter chromosomes gather at opposite poles, and a nuclear membrane and a nucleolus forms around them on each side of the cell. Once the two nuclei are formed, the cytoplasm needs to split during cytokinesis. In animal cells, this involves constriction in the center of the cell. A cleavage furrow forms and the cytoplasm pinches off, dividing the cell into two. In plant cells, a cell plate forms between the two nuclei. Cellulose microfibrils are then laid down on either side of the cell plate, forming a cell wall that separates the two nuclei. Meiosis is the second form of cellular replication and contains two divisions in order to create four haploid gametes. These gametes are combined with gametes of the opposite sex in a process called fertilization that creates a diploid zygote, which undergoes embryogenesis to become an embryo. Meiosis is prefaced by the same interphase stage as mitosis. During the first meiotic phase, prophase 1, chromatin condenses into chromosomes and the nuclear membrane of the cell breaks down. Homologous chromosomes pair up in a process called synapsis, which results in the formation of tetrads. During synapsis, the chromatids of the homologous chromosomes cross over at points called chiasmata, allowing them to exchange genetic material. This results in a recombination of genes in each homologous pair. During metaphase 1, homologous chromosomes align along the metaphase plate, assorting independently of each other. Spindle fibers from the centrosomes attach themselves to kinetochores on the centromeres of each chromosome. During anaphase 1, the spindle fibers contract and the chromosomes move to the poles of each cell. The cleavage furrow then forms during telophase 1, and finally, the daughter cells separate during cytokinesis 1 to create two haploid cells. These two haploid cells then undergo meiosis 2, which is effectively mitosis but with half the chromosomes. The spindles form during prophase 2, chromosomes align during metaphase 2, centromeres are cleaved and sister chromatids separate during anaphase 2, and cleavage furrows appear during telophase 2. In cytokinesis 2, each cell splits to create four haploid gametes. It's important to note that meiosis occurs differently in male and female organisms. Male germline cells are called spermatogonia and undergo the process of spermatogenesis to create spermatocytes. Female germline cells are called oogonia and undergo oogenesis to create one gamete called an oocyte and three polar bodies. At its core, mitosis ensures the continuity of species by enabling growth and repair, which means that an individual organism is better suited to survive until it can reproduce. Meiosis ensures species continuity by enabling sexual reproduction, while mitosis enables asexual reproduction, like in the process of budding. However, these two processes also have numerous mechanisms to ensure genetic stability and to introduce genetic variation. There are a number of aspects of mitosis and meiosis that ensure genetic stability. Telomeres exist on the end of chromosomes, so when the ends of the chromosome aren't replicated, no important information is lost. Error correction during interphase ensures accurate replication by fixing incorrectly placed base pairs. Haploid gamete creation means that the fertilized embryo is diploid and hence the ploidy is maintained. If the ploidy is disrupted, the resulting embryo will either not reach birth or will have disorders. There are two key factors within meiosis that ensure genetic variation. The first is random gamete formation, which relies on crossing over, ensuring that the daughter chromosomes in gametes have a mixture of paternal and maternal alleles, and independent assortment, which ensures that each gamete has a variety of maternal and paternal chromosomes. The second is mutation, which occurs during the synthesis phase prior to meiosis within germline cells. If a base is substituted, deleted, or inserted during DNA replication, a new allele may arise in the gametes. These two sources of variation increase the species' resilience to environmental change and hence aid species continuity. An example of cell replication aiding in the continuity of a species is the development of malaria resistance in humans. The Duffy antigen gene encodes for a protein that the malaria parasite needs to enter a human's bloodstream. A point mutation in this gene disrupts the creation of the Duffy antigen, impeding the parasite. It must be noted that while genetic variation allowed the mutation to occur, the genetic stability that meiosis and mitosis provide has meant that the mutated allele has been maintained along generations to the point that 95% of the West African population have it, and Plasmodium vivax is nearly completely absent in West Africa. In this sense, mitosis and meiosis, which allow for genetic variation but primarily lead to genetic stability, ensure the continuity of species.